The following presentation is part of the Agronomy and Horticulture Seminar Series at the University of Nebraska-Lincoln.
Yeah. Good afternoon, everyone, and maybe good morning or late evening for some of you who are joining today. I'm glad to see a good attendance. It is my pleasure to introduce my colleague and friend, Professor Menahem Moshelion from the Robert Smith Faculty of Agriculture, Food and Environment at the Hebrew University of Jerusalem. I had the pleasure of interacting with Professor Moshelion for maybe 3 years back, and since that we've on and off been in touch, including co-advising an excellent student from his department who was also at Nebraska. So Professor Menahem Moshelion is a molecular physiologist. He has been interested in studying both the molecular and cellular processes as well as the whole plant physiology. Oftentimes we think that the whole plant physiology is very simple. It's the molecules and the genes and the interaction and the signaling that's more complicated. What we all sometimes ignore is that they're all integrated into one big response. And I think Professor Moshelion's done an excellent number of research projects on stress, which is also of great interest, including work on aquaporins, and which are small proteins that regulate plant water status, along with Professor Ronnie Walash, who I also had the pleasure of being introduced when I was visiting Israel a few years ago. He's developed a really cool whole plant phenotyping system that provides a boatload of physiological data. And coming back to my point about the physiology, whole plant physiology being less complex, I think his work's shown that it's less complex if you only look once and at any given time of the day. But if you start to make multiple observations over time, like most biological systems, it's very complicated. So today he's taken some of the advancements they've done with the phenotyping into a successful company which has commercialized. It's called Plant-Ditech. So I think they're doing very well, and I think we're going to learn more about the science and the technology today. So Menachem, over to you. And thank you for accepting this invitation. We were hoping to host you in person, but it is what it is. We are looking forward to your talk. Welcome.
Thank you very much, Kamal. I'm very glad to be here. I wish I could be in person, but this is the status, so we just have to live with it. Maybe next time. So hello everyone. So thank you for this nice introduction, Kamal. So as Kamal said, I'm a plant physiologist, and I really my main study are to understand how plants regulate the water relations in order to maximize yield. Let's talk about this concept for a minute. So when we look on a wild type in nature, we can say that this plant reaches some optimal behavior in its own environment. But when we talk about breeding, so breeding actually put a lot of pressure on the plant in order to get much more yield. Now, when we look on the many years back, the literature, we see that mainly the breeding process increased the yield because it's also increased the size of the plant and the number of stomata, but much less because of its improved the plant efficiency. So plants after breeding process usually are bigger and produce more yield, but many times we see that are also less sensitive to ABA or produce less ABA, which means they open stomata for a longer time of the day. All in all, their productivity is much better. We know they have more yield, but also their vulnerability, in particular vulnerability to drought. And when we look about the reason for that, the reason is the ratio of water to CO2, which means during transpiration, crop plants lose in average 500 molecules of water in order to fixate 1 molecule of CO2. If you look on the whole plant number, to produce 1 banana in Israel, the farmer needs to irrigate the plant about 100 liters per 1 banana. If we talk about avocado, So 1 kilo of avocado is about 500 liters of water being irrigated to the plant. 1 kilo of avocado is like 3 to 4 avocados. So these numbers are huge. Crop plants really take a lot of water, and we can see it in many, many other crops. For example, you can see this wheat breeding program. You can see this is the year of release. So of course, as more recent the year is, the more the yield this crop produces. This is what we expect from a breeding process. But when this the researcher, this Australian researcher, took the same plants, and this time he measures stomatal-conductance, he find even more linear slope, which means the higher the plant produce yield, the higher its stomata are open. Okay, so you can see that the high yielder are almost double in stomatal conductance than the low yielder. And this is mainly due to breeding process. So when we have breeding lines, we can say that the pressure of breeding improve the yield. This is clearly, but mainly because of canalizing many key traits like photosynthesis and transpiration. But this is in the price of less water use efficiency or lower water use efficiency, which mean higher risk, because if this plant opens stomata for a longer time, they are more susceptible to drought. And you can see it every time we have a big drought here. Most of the modern crops can lose up to 80% of the yield because of drought. Similar thing with salinity, but drought is one of the main problems. So obviously many breeding programs are aimed to face this problem of drought and salinity, but the breeding for drought-resistant plant can take many years. You can see it can take more than 2 decades. And the reason is not just because of the normal long way of breeding, finding the right trait for performance. It's also the problem of genetic by environment interactions, which mean every year we have different conditions and the plant respond differently. So all in all, we cannot really expect the same result every year, which in the final result take much more time to breed. So what are we breeding for? There are many terms in literature like drought resistant, drought tolerance, drought resilience. I want to talk about the way I'm looking at this terminology. So when we looking at the plant, usually at the beginning of the season, everything is okay. I mean, usually there is not stress at the beginning of the season. And then when stress comes, the plant can respond either by susceptible, that means it's very sensitive to the stress, it might die, or to produce escape mode, which means plant will flower soon, would not produce too much fruit. But what we are very interested in the breeding process is what happened, what are the mechanisms the plant activating during the stress? What are the defense mechanisms? What are the biochemical, physiological, anatomical defense mechanisms that the plant actually activate? Because this mechanism can, in fact, make the plant recover the stress. For example, longer roots may reach water at lower part of the soil, and then the plant can recover. Or recovery could also come by just rain. Either way, whatever happened during the stress will help the plant to recover. And this recovery process, the terminology is resilience or recovery from the stress. Now we must look at it in 2 phases. The vegetative phase, which means plant biomass recovery, leaf, roots, and so on. And we have the reproductive recovery, which means how many flowers and fruit the plant produce. So if after the stress, the plant is soon to flower, we talk about low resilience and low tolerance. Some plants recover nicely in the vegetative part, so they have more shoot and roots, but don't have more flowers or fruit. So we can say they have high resilience, but low tolerance. And what we usually looking for is high resilience and high tolerance. So plants that will keep growing and also make as much as possible yield. And all have to be compared to the control plant or the resistant plant. What do I mean? Control is a plant that was not exposed to stress, so we can say that it actually uses all this time to produce. So this is what the genetic potential of the plant might be, or resistant, which means doesn't sense this stress. Personally, I don't believe there is a resistance to drought, because plant without water cannot produce that much. I just showed the number. But in other stresses like salinity, they might be a resistance. So this is the terminology I'm going to use here in my talk. So let's go back to the breeding process. Our research hypothesis was that if we go back, that means if we do reverse physiological phenotyping and look on plants that are already past this breeding process and the yield is already known both under normal condition and stress, we might study their behavior and find some cellular physiological or crop physiological traits that could be used as traits for future breeding. So what we did, we took the tomato introgression lines. This is a library that developed by Professor Dani Zamir, my colleague at the university. This is the M82. It's an old breed line that was crossed to the wild type Pinelli. And in each chromosome there of the mother plant, the M82, there is a piece of the father plant, the Pinelli. So in every chromosome, we have one or at least one piece of the father insertion. And we have only one at the whole genome. So the plants are very, very similar, except this single unit of chromosome that were passed from the father. These are stabilized organisms because of many back crosses. So we took this library, and Dani Zamir was very generous to give us many yield results from the field. So we took the field results and we compare all the lines. So there are basically 76 lines. We narrow it down to 30. And we look on the M82. The M82 you can see here in black. This is the mother line. And we checked for the yield under optimal irrigation and drought condition. And also we checked for the plant biomass under optimal irrigation and drought conditions. And we choose plants which are at least 20% higher than the M82, or similar to the M82, or 20% lower than the M82. For example, you can see this line IL1211. When you compare it to the M82, you can see that he had higher yield and higher shoot mass under well irrigation condition. It also had higher yield and higher shoot weight under drought. So this is an idiotype, or we call it high yielder, high biomass, high tolerant, and high resilient plants. This is the abbreviation. We did the same characterization to all the 30 lines. And all in all, we found 20 different behavior patterns. We select 8 out of them, 7 plus the M82. And we chose these 7 first because we want to have very different lines. So for example, we have 1 idiotype, 2 high yielder, 1 medium yielder, and 2 low yielders. The color you see I'm going to use for the rest of my talk. So green and blue are the high yielder, and red and yellow are the low yielder. And we choose this line because we had at least 4 years of field experimental data. So we have to make sure we have a robust data set, because now we want to study the behavior of this plant. So we grow these 8 lines. We started in the greenhouse. This is a protected greenhouse. So the plant growing in the soil. And we start by using a lycra machine, like a gas exchange. But we didn't find any differences. It was very disappointing, neither in assimilation or stomatal-conductance. And when we thought why, we understand that this is because these machines are a steady state machine. So in this chamber here, you have to wait for a steady state to occur before you can take the measurement. But if you really look on a greenhouse, this is the temperature in my greenhouse. You can see every minute, actually, at each point of the greenhouse, the temperature are getting very, very different. And the colors you see are at 2 heights, close to the surface and 1 meter above the surface. So obviously, until you move from plant to plant, all the conditions are changed. So it's very hard to measure a plant under normal sky. You see, this is the light condition in the greenhouse, and this is the VPD, the vapor pressure deficit. So if you just run along with one machine, it's very, very hard to get to significant data. Also, the plants are growing. So if you do this data every few days or weeks, you get totally different microenvironment of the plants. Moreover, usually when you work with this manual machine, you just push the leaf into the chamber, and the leaf assumes that you have 1-to-1 stomatal density ratio between the adaxial, the upper part, and the abaxial, the lower part. This is not a problem if you just measure one plant, but if you compare many plants, you actually must make sure that the density is the same in all the plants, which is not always the case, and I will talk about it more later. So we look for a high throughput phenotyping system, and which at the beginning, many years ago, we start with phenotyping with imaging, but as Kamal said, we develop our own physiological system. I want to say a few words about the system. So this system actually is a physiological based high throughput phenotyping system or functional system. What do I mean by that? We use the system to measure all the plants simultaneously when they are randomized in the greenhouse. So we can really build a truly randomized experiment where each plant been monitored for its soil, root activity, whole plant transpiration and stomatal-conductance, and the environmental. So we measure the soil, plant in few measurement, and atmosphere conditions. So we can say that each plant is like a resistor that we measure its conductance between the atmosphere and the soil. I will explain more about how we do it. So if you take just one plant, the plant is sitting on a lysimeter. It's basically a balance, compensated balance, that was specially developed to work in a very accurate way. We also have all this plastic that help us to measure the plant and to get very accurate data on the canopy, the root, and the shoot. Each plant has its own controller that both measures soil water content with a probe, atmospheric condition with the sensors, and regulate the irrigation for each plant separately in a feedback way, which means I can irrigate each plant according to its own performances. This helped me a lot when I try to regulate complex traits like drought. Now, lysimeter are old technology, and basically just the old lysimeter just measured the whole plant. An old lysimeter is like a normal human balance that measures the whole weight of the body, but cannot separate fluids for fat, for bones, for example. So what we did, we actually, in our controller and algorithm, we canceled the use of data loggers, and we could separate the shoot from the root, from the soil, and from the water. So basically, we can measure separately shoot conductance, root fluxes under different soil conditions. The measurement we take with the system is whole plant biomass gain. So we can measure the continuous growth of a plant. We take transpiration, water use efficiency, stomatal conductance of the whole canopy, root fluxes, and we can also identify the drought point, the physiological drought point, which tells us exactly what point the plant becomes under stress. More importantly, all these measurements are absolute numbers, which means we have the whole plant in a very simple numbers like ml per plant per day. So we can really know the exact amount, which usually we cannot do when we work with Leica, for example. So it's only for one leaf. When we work with a pressure chamber, it's one leaf and also a relative number like water potential. But with the plant array system, we can really get the whole plant absolute number. So for example, transpiration could be ml per plant per day, how many ml the plant lost. We also have tools of analytical tools that we can get the raw data, some basic statistics like ANOVA and t-tests, box plots, histogram, and also a heat map that we can really identify each plant in the array. And if we need RNA or DNA or something like this, we can just approach the plant. Very importantly, the system helps us solve many of the pot effects. You probably know that when you grow in a pot, you may very fast be exposed to the pot effect. For example, big plants will transpire much more than a small plant. That means that a smaller pot size will make your plants to get much faster drought, which not always what you want. Or you get a lot of variation between different experiments. So the system helps solving this problem of the pot effect. I'm not going to get into too much details, but this was published in this paper if you want to read more. Also, if you want, I will be glad sending you this paper explaining exactly how this pot effect is solved. So all in all, the system enables us to control each plant separately to any nutrition and water level we want. We have all the plant measurement simultaneously and continuously, so we can compare all the plants to the exact identical conditions. And we have an analytical tool in real time, so we can very quickly compare these plants. All the algorithm was published in this plant journal, Technical Advance. And if you want to see the canopy conductance, which I'm going to talk about more today, this was recently published from a group in Wageningen University that actually tested our stomatal-conductance accuracy, and they found it's very good accuracy. So I'm not going to get to too much details, but if you have questions later, I will be happy to answer, or you can see this paper for the answers. So let's go back to our lines. So we choose these lines. We run them on the plant array system. Let's look a bit on the raw data so you can see what we measure. So this is the raw data of one plant through the whole experiment. You can intuitively see that the plant is growing with time. Let's focus a bit to see what exactly we see. So this is the weight, this is the raw weight. The increase in weight means irrigation, and the decrease in weight means transpiration. So when we define the RDT morning and RDT evening, the delta between the two, the line here, is basically the plant water loss during one day. So if we just want to see how much the plant loses, we just need to take this delta every day. And the line here shows us a continuous plant transpiration increase. Usually it's because of the plant is growing. Now, this is the IL81. To remind you, this is the low yielder, you see LY. And this is the daily transpiration of this plant during well-irrigated condition. This is about 2 weeks or a bit more than almost 3 weeks of well-irrigated condition. Then we expose the plant to drought, and then we put it to recovery. Okay, so we bring back the irrigation. This was the experimental scenario. And of course, we compare to the all other plant. Each line is the average of at least 5 different plants. This is the 5.2. You see, this is the high yielder. You can see that this plant has much faster development of daily transpiration with very fast decrease of transpiration during drought and very nice recovery. And this is the M82, somewhere in the middle. Okay, this is the mother line. Of course, we compare all the lines together, and you can see that if we start experiment when the plant were 4 weeks old. So you can see that at this age, no significant difference occurs, but in few days, significant difference start to occur, and the high yielder, like 11.4 and 5.2, really become very fast, like in about a week, become significant from the low yield. So when we expose them to stress, you can see that the high yielder reduced transpiration very fast compared to the low yielder. We have a transpiration flip-flop. But this is not because of fast drought, because if you look at the drought, this graph here shows you the soil water content. You can see a similar slope that we could regulate because of the system. So all the plants actually exposed to the same drought, but this plant respond faster. Okay. So I will talk sooner about how can we regulate this drought so accurately. But let's start, before we talk on the drought, let's talk about normal irrigation. So let's look at the window here. When the plants are well watered, all of them just get full irrigation. When we just look on the daily transpiration, so this is the number, this is the gram of water plant lose in one day. But it doesn't give us too much resolution. So if we want to look on higher resolution, what we do, we take the momentarily resolution of the plant. So let's see how we do it. This is the light in the greenhouse. This is the VPD, vapor pressure deficit. This is a very important parameter of the atmosphere. It's taking account the temperature and relative humidity. As higher the temperature, the more demand the atmosphere put on the plant, and the plant actually start to transpire more. The black line you see here is the weight. This is what we get with the lysimeter. So you can see like 600 ml decrease in weight of 1 day. And when we take the first derivative of this weight, we actually get transpiration rate. So this is millimole per mass per second. So this is the transpiration rate of the plant. And if you want to take the stomatal conductance, one of the most important parameters, basically is how much water the plant loses normalized to its weight relative to the VPD, to the demand of the atmosphere. And this is the green line. So this is the stomatal conductance. All of these parameters we can get directly from the system. You can see that the stomatal conductance is open wider at early time of the morning here when the VPD is low and the light is relatively high. So let's look on our plant. Let's start again with the 8.3, the low yielder. This is the stomatal conductance. You can think about stomatal conductance as how much the stomata is opening, right? It's the conductance of the stomata to gases. So we can think of it as stomatal aperture. You can see it's respond very nice with the light and VPD. When light and VPD goes up, also the stomatal conductance goes up. And when light goes down at the evening, this is evening time, this is the time of the day, also stomata start to get closed. Look that the VPD goes up at the same time. This is because of other atmospheric conditions. But again, the stomata getting closed. So although the VPD goes up, the stomata is responsible to the light. You can very nicely see this. But when we're now measuring the actual transpiration rate, which means how much water the plant really loses, you can see that while the stomata is getting closed, the transpiration is staying fixed. And this is because of the higher demand. So when the atmosphere demands more water, although the stomata getting closed, the transpiration is still fixed and only later it's getting closed. This is very nice difference between transpiration rate, water loss, and stomatal conductance. Very key important physiological parameters. Now we compare all the plants together under well irrigated. We pretty much see the same pattern we showed before. You see blue and green, the 5.2, and 11.4, they are big here, and also they have high transpiration here. In the stomatal conductance, we see less big difference. I mean, the 8.3 is always the lower. And there is also very interesting thing here with the M82. You see, it's open its stomata longer time in the day. This is because this plant was a past a breeding process. Remember, I told you that breeding process usually push this stomatal conductance longer during the day, and you can clearly see it here. But all in all, under normal condition, we don't see something different than what we already saw in the daily transpiration. What we really want to see is what happened under this stress condition. So, as I said before, during this stress, many mechanisms being activated, and we want to know what happened here. So one simple way to see it is just follow the recovery rate. So after the drought occurs, we can say that during the drought, all this mechanism of defense mechanism being activated. Now, it's very hard to see them because usually stomata is getting close, so it's very hard to understand what happened to the biochemical, anatomical differences between the plants. But one very nice way to look at it is the recovery rate, because if the plants could survive the drought very nice and recover fast, that means that it did something good here. But if it could not recover after the stress, that means that it suffers somehow from the stress. So one way is just looking at the recovery rate and compare the different lines in the recovery. What we do usually, let them go back to the recovery to pretty much what they have before the stress. This is the resilience. The slope here is the resilience. And when we look on the recovery rate or the resilience, we can see that the 5.2, this line here, and the 11.4, these two lines, so they were the higher transpiring plant, but also fast recovery plant. So although they transpire more and close the stomata sharp, they could also recover fast. You can see it here, at least compared to the 8.1, the low yielder. Also, we finished the experiment here. So we took the plants and took the dry weight of the plants, and we compared the dry weight of the plants. So actually, whatever the plants could fixate during this period, we took it as a dry weight. and we compare to the cumulative amount of water the plant transpires. So we just took everyday transpiration and sum it up. So the graph you see here is the cumulative transpiration versus the shoot weight. And very interestingly, these two lines not only transpire a lot, but also get a lot of biomass. And they did it very efficiently. When you compare the biomass, the dry biomass, to the total water, we call it agronomic water use efficiency. You can see that at least the 11.4 is significantly more efficient than the 8.1. So not only it transpire a lot and produce more, it's also do it more efficiently. So these are three important characteristics that you can just easily take. So more importantly is what happened during the stress. What does the plants do during the stress? So first of all, when you compare all the plants under stress, you must make sure that they are really facing a similar stress. What do I mean by that? So this stress here, if we would not regulate it very efficiently using the planetary system, we will get this. This is the same IA lines when we just let them dry. When we just close the tab here and let them dry, the fast transpiring plants lose water very fast, while the low transpiring plants lose water much slower. So let's say after a week, the high transpiring plants will be under much severe stress than the low transpiring plant. So you cannot really compare them based on time. So this is why when you want to compare apples to apples, you must make sure that all your plants are under similar stress level. Otherwise, you really compare the time and not the stress. So how can you determine the stress level? So the planetary system have a very unique controller with two valves, A and B, that first of all can make a cocktail of solution, but more importantly for this experiment, they can irrigate the plant based on its own previous day transpiration. So if a plant, for example, loses 100 ml and you want to expose it to 90% drought, you will just give him back 90 ml. And because each plant has its own controller, you can see it here, we can regulate the drought for each plant separately based on its own physiology. We can also do automatic dehydration rate. So we can just do what you saw before. You can just get the drought in a very similar rate as I showed you before. And this drought is basically what I showed you before as you can see here, as a very similar rate. Now, when we control some people use the system can also control the drought using a sensor. What do I mean by that? The system can be hooked to all kind of soil sensor and regulate the water based on a sensor. But this could be sometimes misleading. Let me explain you why. Let's look on these two plants, tomato and sunflower. You can see both transpire the same and the x-axis here is the soil water content, measuring by a probe. Now, the tomato plant, when we start dry the soil, will close stomata at around 60% of soil water content. This terminology for this is theta-crit. Theta-crit means the drought point or the soil water content, which become limiting factor for the plant transpiration. This is a physiological point that actually under this point, the plant been exposed to stress. At the meantime, sunflower is only being limited under 40% soil water content. This is because of the sunflower root system. So if I would regulate the drought based on a soil probe, for example, let's say I would choose to put the soil on 50%, it's not a big deal. It's very easy to do with the system. But in this case, without knowing the theta-crit of each plant, I might put the tomato under stress while the sunflower won't be under stress. Okay, so controlling drought treatment based on soil probe could be misleading. The reason, by the way, for the sunflower to have a lower theta-crit is because of its much larger root system that actually reach more water in the same pot. So although both of them exposed to the same depletion of water, sunflower can reach to more water and actually reduce its theta crit to a lower values. So in our case, we took the theta crit to each one of our plants, of course, and we choose the drought point to be below theta crit at about 50% of the transpiration rate of the whole plant. So we choose the stress to be physiological stress level. Now, again, we compare all the plant continuously under the same light and VPD conditions. So let's start with stomatal-conductance. You can see again that the stomata is opening. First of all, the values are much lower than what I showed before. But also you can see that although the light and VPD are pretty fixed during the noon, the conductance is getting lower because this plant is already exposed to stress, and this is because of ABA. But when we compare the 8.1 line to the 5.2, we can see very interesting result. You can see that 5.2, this is the high yielder, we call it Idiotype, has much faster opening of stomata early time of the day, and later on close the stomata, getting much lower water loss. So it can open stomata wider, meaning getting more CO2 at this time of the day, while paying a lower price of water loss. You can see it here. So this is a very nice difference between the high yielder and low yielder. Wider stomatal conductance, at the same time lower water loss because of the different time of the day and the different VPD. The VPD, as I told you before, this is the demand. This is what actually pull the water out of the plant. So of course we compare all the plants simultaneously. You can see nice patterns, a bit different between the lines, and we were wondering, why is these differences? I mean, how come these different plants, all of them look almost identical, the tomato plants? Why do we get these patterns? And we saw this paper by Henry et al. That talk about stomata safety efficiency trade-offs. So they claim that the difference in between the stomatal density and stomatal aperture might have a role in the response of the stomata to the environment. And we were wondering, could it be really that the stomatal density is different between our line? So what we did, we used this special stomatal printing method. It's a special glue that you can just fix to the leaf. It's very fast dry, so you can get the printing of your leaf. And we want to have both the abaxial and adaxial, and we want to have this printing at different times of the day, at least 6 times of the day to all of the lines. To have this simultaneously, you really have to have an army of people running around in the greenhouse and gluing this material. By the way, if you want to see the protocol, we just loaded to the bioarchive. It's now under revision. And what we could take out of this measurement is both stomatal aperture and stomatal density, which means how many stomata we have in 0.1 millimeter. Now, when we look at the results, and of course, we did it both to the abaxial and adaxial, the upper and the lower part of the leaf. So if we look on the total density, we don't see significant difference between the lines. But if we look on the difference between the abaxial and the adaxial, we see that in 5.2 and 11.1, these are the high yielder, there is much more stomata on the lower part of the leaf than on the upper part. Some plants are neutral, so they have the same. And some of the low line, like 8.13, also have similar to the high yielder. That means that this is not enough to have this difference in densities. So what we did, we also look on the aperture, okay? This time, a different time of the day. So we look at the aperture at different time of the day, and you can see that 8.1, the low yielder, has its maximum aperture at the midday. Now, this is Israel. Midday is very hot. So opening stomata to its wider peak at noon might be not the smartest thing to do, especially when you look at the 5.2 and you see the peak is really at the same time we saw the stomatal conductance peak. So relatively early in the morning at the lower part of the leaf, and in the upper part, it's a bit later, but also before noon. And the MET2 is somewhere in the middle. And we did the same to all the plants, and we find very interesting trend of maximum peak of the stomatal aperture. We were wondering if this density differences is related to the speechless gene. Speechless is a gene that involved in the regulation of stomata in the leaf. And we also were wondering if some of this time of the day is related to some stress gene like this zeaxanthin epoxidase, which is related to stress. So we took the Chitwood paper that actually take the same library of tomatoes and measure these genes. And we saw very nice correlation with the speechless to the abaxial side. So speechless is very nicely correlated with the stomatal density at the abaxial side. So the gene express much more where we see much more stomata. And at the adaxial, we have much less. So at the lower part, we think that the speechless is very much involved. The ZEP1, the zeaxanthin, just show us a mirror image, which means that the 8-1, although it was not under stress at this condition, show high level of this gene, which is like this plant is under constant stress. This is like what seems from this gene, again, with a very nice correlation. So to sum up this part, when we measure the aperture is very important trait, very hard to take, but it's very important. But measuring the aperture of the stomata is not enough. You also have to know the density and distribution of the stomata between the adaxial and abaxial. You have to know your VPD, which is the temperature and humidity at every time of the day. And only when you have these three, this actually sum up to what I showed you before, the canopy conductance and the transpiration rate. So we saw very nice fitting of what we took manually to what the system gave us automatically. Now, of course, when we come back to the manual machine, if you really want to measure it manually, you have to work with probably a few machines and do a very tedious work. People did it. You can see this very nice work of Broderick and Holbrook showing the same, we call this peak, by the way, golden hour. Seeing this golden hour, different lines, again, as we saw before, when the VPD is low, the stomatal-conductance is high. They did it here manually. And they could also show very nice increase early in the morning. Some plant almost totally close their stomata at noon. We also measure it in other paper we show with the lysimeters, the same high peak at morning. Again, it was highly correlated to yield. Another work by Bachar et al., a student of Professor Peleg and Arkamal from your institute, show the width cultivar introgression lines with better yield, better resilience, also had this nice peak in the morning. So we can see these trends coming from several plants. So to conclude this, my main take-home messages. So high transpiration is a very good trait for yield. We saw it in all the high producing lines, very high transpiration. But under drought, a fast response or plastic response of the stomata is also very important. So as faster the plant responds, the better. High abaxial to adaxial stomatal density. So the more stomatal you have at the lower part, the better. Dynamic water use efficiency, in particular, the golden hour, it's a very nice trait to stress response. So the plant that open stomata wider and under stress earlier in the morning is better. So we call it dynamic water use efficiency. And rapid recover, what you see here from stress. So also, if the plant transpire a lot and it's really rich high transpiration before stress, during stress, it's probably exposed to more stress because this plant is physically bigger. But if it can still recover fast, this is a very good trait. This fast recovery, it's a very important trait as well. As I show you, we saw that the speechless gene is probably important in regulation the gene expression, and the zeaxanthin also important there, probably as a stress response. So obviously, if you can measure the stomatal aperture is very important, but both manually, as I show you, or using steady state gas exchange is very tedious and very low throughput. What I show you is the planetary system that you get, first of all, the whole plant measurement, absolute measurement, and you compare it continuously and simultaneously. So we think that in breeding or a similar functional project, this is very, very important. What next? So we are now developing several simulations with many other functional phenotyping tools to help us actually breed to several scenarios of stresses, salinity, nutrient, and so on. And the idea is like choosing the winning horse based on its real race and not based on its genetic or other measurements. So we always compare our plants together. When you choose a phenotyping system, actually, as you know, there are many phenotyping systems. What we recommend is if you're doing a stress response, you should take in account these 10 key challenges. First of all, remember the plant responds very fast to the environment, so you better measure the shoot, the root, the water in the soil, the atmosphere, light, VPD, and if possible, also the rhizosphere, because all of those traits are cross-interacting with each other. It's better to measure several traits like stomatal-conductance and transpiration and root fluxes, biomass, again, simultaneously under well-irrigated and drought conditions. Continuous and simultaneous measurements are crucial because of these momentary changes. If you really want to see the difference, you must know they are really simultaneously measured. You need to, if you're doing drought, it's better to control the drought and not just let it dry. Also, probe-based regulation is not always good. It's good, but you have to know what you're doing. So standardization is very important in order to repeat the experiment again and again. Solving the pot effect is crucial. Smaller pot will just make the plants dry much faster than you want. And working in a randomized way, so the controller helps us actually measure all the plants simultaneously by controlling each one separately. So truly randomization and real-time analytical tool are also very, very important. I don't have time to talk about two other functional projects we did, like biostimulant and early detection of plant disease. So I will just skip it. In few words, we could screen for biostimulant effect. So you can use the same genotype, but use different chemicals like biostimulant, again, using the same principle and really understand the difference between different chemicals. You can really understand what they are doing. This saves a lot of time before going to the field. And recently we start to work a lot with biotic stresses like plant diseases. This is in collaboration with Shai Kobo, my colleague. For example, this fusarium that could very easily show phenotypic, I'm sorry that I'm jumping, but I don't want to take more time. You can see that we can see differences in transpiration of infected plants after 5 or 6 days, while to see it in your eyes, it takes like 3 weeks. And the more severe the stress is, the faster you can see the differences. So biotic stresses like this fusarium or, sorry that I'm jumping, but we did it in several, or these viruses also on the lead system, you can see differences in the plant, whole plant behavior much before you see anything with your eyes. So we think this functional phenotyping could be also very nicely implemented in biological stresses like diseases. I would like to thank Sanbon. He's the PhD student, did most of the research I showed you. Professor Dani Zamir that gave us all the IL lines and all the field results. Bogale, Ravi, and Ramon, master student, that's also a part of the paper, and Shani Friedman that did the disease research that unfortunately I could not explain it too well. And thank you very much.
Thank you, Professor Moshelion. That was an excellent and very insightful talk. So there's a question. Thank you for the very nice presentation. I'm curious about the significance of calling these recombinant bread lines idiotypes.
So we chose this name because maybe I will share again. We chose this name because of this table here. So basically, when you look at this plant, we compare them always to the M82. Okay, M82 is the breeding line, so it's like the line that was released. But these plants were higher in yield, higher in shoot weight, higher in fruit under drought, and higher in weight. So actually, they were better in all measurements. So this is why we call it idiotype. It's just something you can say we decided to call it.
Another one. Have you, by chance, compared results of idiotype assessments against foliar damage following lethal drought treatments? If so, did you see strong correlations? And Aaron is asking this because I have an interest in plant survival in wild populations.
Yeah, it's a very good question. So yeah, we did something similar with barley cultivar. Let me find. It was actually published. So we collected barley from different zones in Israel, from the south till the north, and we compare the barley behavior. This is wild type, exactly for this kind of behavior. We didn't put it to a little stress, and I explain why. When you work with ecological stress, so what we did, we actually took 6 years back of these regions, and we measure what is the average time between 2 rain events. Okay, we call it drought duration. So average drought duration was the time period between 2 rain events. We also took soil samples and calculated the water content in the soil, and we built in the system 2 simulations of severe drought and less severe drought to all the barleys together based on the actual ecological data that we got. So we are building the scenarios based on what we expect the plant should be exposed to in the field. So usually we never just close the tab and let them die, but try to keep the stress to be similar to the desired scenario.
Hey, yeah, thank you for the presentation. Yeah, I'm curious, after seeing the slide with like your variation in stomatal kind of peak opening and transpiration, those points in a controlled environment situation, do you think we could manipulate light intensity, VPD, to kind of capitalize on staying more at that peak level during the day?
Yes, I'm sure we can. We do it with cannabis plant. Well, you cannot fully control it, but it's for sure much more easy. And this peak is really not the morning. The reason it's in the morning, because this is the right combination of VPD and light. But if you can mimic it, for example, if you take the same tomatoes during the winter, the peak shifted toward the noon in Israel because you have much less light and all the other condition changes. So this is really depend on the environment, not exactly the time of the day. So I think, yes, I think that in a controlled growth room, you can just follow it and each time try to improve it based on the plant feedback.
Looking at the transpiration rates, is that what you would look at as a metric for that?
So I would compare both transpiration and conductance. So transpiration is the actual water loss. In the growth room, it might be enough because usually you can control very nice the VPD and this is the main thing, the VPD. But both, I would look both on the conductance and the transpiration rate.
Great, thank you.
There's also a question on about identifying any genes that you think could be associated with some of these phenomena. You mentioned speechless.
Yes, I mentioned. So we didn't do the genes. We took it from a known library. So this IA library has many papers on it. One of them was this expression profile that they did. So we found the speechless, which is taking part in the regulation of the stomatal density, was highly correlated with the high number of stomata in the lower part of the leaf. We didn't go too much to it because this is more physiological based study, but these 2 genes that I show you was really nicely correlated. So of course there are probably more genes that are involved.
So do you think that the response of stomates, especially for the high yielding, like the steeper decline under drought, and then of course better recovery, intuitively you would think that they're related, right? There's same phenomena of more responsive stomate, but have you seen any lines where they are actually disconnected?
Yes, yes. So we saw it with rice plant. We talked a bit before about it. So rice are highly productive. They have huge amount of transpiration, so they're very nicely productive. But at least the lines we checked, it was Japonica and risotto, I think, they had a very bad recovery rate. So they had very nice transpiration and fast increase in transpiration, but very bad recovery. This is one example I can recall, but I think it's different rates. I mean, I think many times it does correlate, but it's not necessarily so.
Does time matter to get results? My question is, how long does it take for stomate to stay open?
Stomate respond very quickly. So in a few minutes, it will change. So if you look on the continuous, this is, for example, you see, this is like 2 hours. So from I don't know, 6:30 to 7, you get very fast response, and then a very fast response down. Now you can see all these curves up and down. This is an average, but it's all respond to these changes in the environment. This is naturally occurring changes because it's a non-controlled greenhouse. So this is the stomata response. You see the transpiration is much more smooth, but the conductance itself, it's much more jumpy. Now in the literature, you can see that the stomata responds in a few minutes. There are all kinds of papers. Some talk about 5 minutes, some talk about 15 minutes, but it's in order of minutes, this response.
How can the plant array, or can the plant array system differentiate the transpiration and the evaporation in early development stages? In other words, is there a threshold?
Yes, of course.
For sensitivity?
Of course. So yes, it's much harder. So it's called signal to noise ratio. So what we recommend is to work with about 80 or 100 ml. It's not too much. You can look at this tomato. You can see they start transpiration from about 50 or 100 ml, and it's go very fast. But if you work with the seedlings or very small plants, like we work with our Arabidopsis, so we just put more plant per pot. You don't have to put single plant like I show you here. In fact, what you can do, and this is what we do also, if we work with wheat on a very early stage, what we do, instead of putting 1 plant per pot, we can put 5, 6, or even 10, just to reach this higher level of signal. So the signal to noise will improve. And we also reduce the pot. So for example, our Arabidopsis, we put 4 to 5 plants in 1 liter pot. What you see here, this is 4 liter pots. And we also have, if we want to work with much bigger plants, let me see if I have it here. We can also work with 25 liter pots. So the idea is just to play with the number of your plants, just to get the minimum signal that you need for the measurement. But it's just a matter of pot size and number of plants in a pot.
So the data you showed had many, the vegetation was quite extensive, and they were also very close to each other. Right. Have you looked at the effect of a set of isolated pots, like they're distant apart, so you don't have more of the canopy dynamics of VPT and temperature playing, as opposed to in a isolated versus like in a canopy? Have you seen those differences?
So of course there are differences. Let me show you 2 conformations. This is the dense conformation. We choose this because this is really simulation of a greenhouse. If we run it a bit, you will see the plants getting pretty big. You can see it here. So they really this is the density. They're really going to be in the field or in a greenhouse. This is how they're going to look in the field, very crowded. So of course you get, so there is no noise or something, but there is a difference of cross interaction because of shading and humidity. But this is a real life situation. When we work with diseases, you can see it here in this greenhouse, we put it much more apart. You see, here they are much more apart, but this is very functional because we didn't want them to contaminate one another. Of course, the plant, when it's more exposed, you will get more transpiration and less impact of shading. Again, this is part of the scenario you need to design because it's very easy to move this system. So you just need to know what is your scenario and just design your table accordingly.
When plants cope with drought stress, how is the fruit number per plant changing?
Yeah, this is important question. We need to separate this. Okay, most of the crop plants, what we do, we judge them by two different ways. One is the vegetative growth, what I show you here today. It's growth rate, transpiration, conductance, water use efficiency, and so on and so forth. This is till you have flowers. Okay, so this is very important part of the growth period where the plant establishes its factory, its leaf number, its biomass. Now, as soon as the plants start to flower, this is the reproductive phase. And here it's totally different questions. For example, some flowers are sensitive to the drought and just fall down. Maize is a great example. Maize is a C4, pretty resilient plant, but highly sensitive to drought. Why? Because of the flowering period. So if a maize plant will face the drought while it's flowering, the chances he will lose most of the yield. Although the plant will look nice. So we have what I called before high resilience, low tolerance. Big plants without too many fruit. So when you test your plants, you need to separate this vegetative part from the reproductive part. On the system, on the Plant-Ditech system, what I show you, we usually measure the physiological parameters, which are extremely important during the vegetative part. The reproductive you have to use different kind of tools because it's mainly flower, pollen, and so on.
I think please join me in thanking Professor Moshelion. It's been a really thought-provoking, seminar, and a good lot of questions and discussions. So we really are thankful for this opportunity to interact with you and learn about your work. So
Pleasure.
Thank you very much.
Thank you all. Thank you for inviting me.